Good morning, everyone, and welcome to the Elixir Czech Republic Friday Coffee, where we are presenting tools and services which are provided by the infrastructure. Today, we have a guest from the Charles University, Martin Pospíšek, and he'll be talking about IRES, IRESite, and HCVIVdb. They are the databases, and IRESite is an internal ribosome entry site. So let's suppose that you probably don't know anything about it, but in case you know, so you will probably learn a lot more than you had expected. Martin, please, the floor is yours.
Okay, thank you for introduction. It's my great pleasure to be here and share information about our databases and tools. And I would start. This is the list of everything I would like to talk about. First, I will talk about the biological background and motivation to create the databases and to prepare the tools. And then I will talk about all the these two databases and about the new tool we use, and I will also show some applications just to let you know how we use that and how it can be used in the real science. So first, as I said, I will talk about the background, biological background and motivation. I would start with very ancient slide from Francis Crick, and as you can see here, it's a schema about the flow of information, about the transfer of genetic information from DNA to RNA to protein. And we will talk here about the transfer of information from RNA to protein, which is called translation, and where the protein is synthesized according to the template of RNA. And basically, we will talk only about the most controlled part of it, about the translation initiation. This is kind of a busy slide. Don't be afraid. I will really simplify that immediately in the next slide. But this is just to show you how the translation initiation in eukaryotes looks like. And for the next talk, it will be important to know that there is a mRNA which serves as a carrier of the genetic information to the translation process, and this mRNA has on one end, which we call 5' end, has a structure which is capped. Here is the structure. This is the chemical structure of the cap. Then there is the coding sequence and some other stuff, and at the end there is a poly A tail. The other end is 3' end. And then please, you should remember that there are ribosomes, which are the machines synthesizing the protein using the mRNA as a template, and the ribosome has small and large subunit. And then there is quite a lot of translation initiation factors, which I am not going to talk about them. And to make a simplified view, we can simply draw the mRNA as a rod, then poly A tail as some small waves, you know, and then we also need to define the 5' end, which we can display as a cap. As I told you, this is the important structure at a 5' end. If we go more to the details, then we can draw a start where really the sequence coding for the protein starts. There is an initiation codon, AUG, And then we can go to the stop. There is the stop codon, 1 of 3, and the coding sequence coding for the protein is only between the start and stop. And between the cap and start, there is a 5' untranslated region. And between the stop at the end, there is a 3' untranslated region. And we will talk here during this talk only about the cap, this 5' untranslated region, and the start. It's important to know that in eukaryotes, the general translation starts by the small ribosomal subunit, which with help of all the translation initiation factors recognize the 5' cap at the mRNA and bind to the cap with all these factors, and then scan from the cap to the start. I will go back, just show you again. Bind to the cap, scan the 5' UTR to the start. And again, yes. And if there is a correct start, the IUG, the start codon, then if it's recognized, then the large ribosomal subunit comes, bind there, then all the translation initiation factors are released. There are coming some other factors. I'm even not putting them down there. And the complete ribosome is scanning the coding sequence and synthesizing the protein. This is basically what we need to know now for the translation initiation and the translation. And because all the translation starts by the binding to the cap, we call it cap-dependent translation initiation. And then we can define what makes mRNA an efficiently translated template. Basically, there should be fulfilled this condition: short unstructured 5' UTR, which could be till 200 base nucleotides. Let's put base nucleotides, no bases. There should be no hairpins, no structure at the very end to the 5' end to the cap, or close to the surrounding to the start to the AUG codon. And then, of course, it's good if around the AUG codon there is some sequence which is called Kozak sequence. It looks like that, the signature, but I'm not going to talk about it. The important is the name Kozak, because it's called by, for this talk, it's called by Marilyn Kozak, who discovered the signature around the AUG. And of course, it's important if the mRNA has a long poly A tail. Here is some results from some previous Kozak's paper, and you can see this is the gel with proteins, and you can see that in this mRNA there is no structure. You can get a lot of protein. If you have a structure close to the 5', you get no protein or almost no protein. If you have a long 5' UTR and a structure close to AUG, this is the lane 4, again you get no protein. I am not going to the combination, but what you can see here, again, if you have a structure at 5' UTR, if you have a long 5' UTR, then you can markedly decrease the efficiency of translation initiation. So this is again the picture we know about the cap-dependent translation, but then there is a question: how can be translated viral non-capped mRNAs? Many eukaryotic viruses actually they acquire the cap somehow to the 5' end, but some of them not. And how their mRNAs can be translated? Also, there is a question: how can be translated mRNAs with long structured UTRs or bicistronic mRNAs and so on? And also, how can be translated mRNAs during various stresses, during viral infection, when all the general translation is really decreased. And this is not functional for CAP-dependent translation. So then we need CAP-independent translation. So kind of translation which is not dependent on the structure on the 5' cap. And the first report about that was first CAP-independent translation was discovered in polioviruses or EMCV virus, and the region which was responsible for this CAP-independent translation was called IRES, internal ribosome entry site. So the ribosome is not binding to the cap, but is going directly to the mRNA close to the start of the coding sequence. And it could be the complete ribosome, only the small ribosomal subunit, with or without factors, or with some other accessory proteins. We will not go to the detail, but just for now, we can look at that, like the ribosome is going, or any part of it is going directly to the start. So it's not using the binding to the cap, and it's not scanning from the cap to the start. No, again, it's going internally to the mRNA. So why it's called internal ribosome entry site, IRES. And these are the structures of viral IRESs of the families, and you can see that the IRESs are quite large in many viruses. They are highly structured. So you have learned now that these are not translated well by scanning from 5', and there should be some mechanism how to allow ribosome to go there internally and to start somehow translation. And please look at this one. This is the internal ribosome entry site from the hepatitis C virus. We will talk about it later on as well. But when people describe many or quite some IRESs in viruses, then people are thinking about how the cellular RNAs are translated during the, as I told you, in cases you have a long structure 5' UTRs, or the mRNA is specifically translated during some stresses, viral infection, and in any occasion when general CAP-dependent translation is decreased. And there was a kind of boom of finding new and new IRESs, cellular IRESs, viral IRESs, and so on. And all these IRESs were really different. Many of them have different structures, or people claim that only sequence is responsible for binding the ribosome and so on. So the important thing is to remember that IRESs by itself are defined only by their function, not the structure. You can have different structure which makes the same or similar. So basically, any sequence structure on mRNA which can function as a something to bind the ribosome and to allow the ribosome to start a translation, except the cap, omitting the cap, can be defined as IRES. And there was a boom of many IRESs found in a literature. And then Marilyn Kozak, that's the reason I was talking about it, because she discovered the the sequence around the AUG, which is found by the scanning ribosome. Then she wrote in the reputable scientific journal Molecular Cell Biology, she wrote a mini review paper where she challenged most of the important experiments and most of the IRESs found in that time. And she entitled the mini review by the title "New Ways of Initiation Translation in Eukaryotes?" ? So that means, are there really new ways? And there was quite immediately reply to that, and many, almost most of the important labs, most of the important authors in the translation initiation field wrote a reply to that, because she was really strict and challenged a lot of experiments. And there was signature of many of these important people in the field. And finally, she got a chance to reply again. And at the end of her answer, she wrote, "It is obvious that the organizers worked hard to collect all those signatures, but to what end? A single voice suffices to present a logical argument. I might be alone in refusing to believe a story with so many flaws, but that doesn't mean I am wrong." And then she touched a little bit politics of the time. Counting the votes determines the answer in politics. Florida accepted. If anybody of you remember the battle in election between George Bush and Al Gore in Florida, but not in science. And then started somehow another era in IRES research, when many people started to re-evaluate the found IRESs, try to find if there are any experimental flaws, if there are really IRES, or if the measured data are because of something else. I'm not going to the details. Can be cryptic promoter, cryptic splicing site, anything else can give you, by the tools used to determine the IRES, can give you the false positive result. And there was a lot of papers around that, proving, disproving IRESs. Basically, after a few years, even many of those people who signed the previous reply or comment to the Marilyn Kozak paper said that they wouldn't have signed it again. And many IRESs were disproved as well. And in that time, we decided to help to the scientific community by putting all the information about the known IRESs in that time together, and and we put that together and made the IRESite database. And the database contained a lot of information about the IRESs, about the structure, but also about the experiments where these IRESs were used, also some kind of evaluation and so on. It was published in 2 papers in the database issues of Nucleic Acid Research, and is quite well referenced till now. And people are using it. And I would like to mention here Martin Mokraj, who was really doing his PhD in my lab and shortly as a postdoc, was really working a lot in this project. Then we are more focused on hepatitis C IRES, which I showed you before. And I would like to tell you that you probably know that what does it mean, hepatitis C? But hepatitis C was discovered in late 80s as a blood-borne non-A, non-B hepatitis, and you should know that there is some acute phase, but it's a really important disease because most infections go to the chronic disease, and in many of these cases, after years, the hepatic cirrhosis can develop, which can lead to the hepatocellular carcinoma, and in many cases to death. And the hepatitis C virus genome is plus RNA genome, and at 5' end there is a structure of the IRES which allow ribosome to entry directly to the genome. There is no cap at the 5' end, and it's important to say that hepatitis C IRES is the most investigated IRES at all. So if somebody would like to investigate, to study really nice small details about the really details about the IRES, how they function, hepatitis C IRES is a good choice. And because there is a lot of mutations about that, so we prepared another public database, and we publish it, which is called Hepatitis C Virus IRES Variation Database, and we put together all the with the experience of our IRESite, all the known mutations, either natural occurring or engineered, and also we put together the experiments and try to somehow evaluate the activity of the IRES if there is this mutation or variants. And this is one of the page of the HCVIVdb. We can, for example, open the domain 2b. Then we can see the mutations. We can see if there is a translation information about this mutation or variant, the publication where it is. Then we can go directly to some entry. We can look on the entry, see can see what kind of mutation is there, the domain. Then if it's natural or engineered and so on, we can see if the same variant, if at the same position are other variants, and what are the publications there, and so on. And I can go through that. This is another entry, and then we can compare translation efficiency of the same variant or at the same position from different publication. We can discuss later on if you want these discrepancies. So the application is what we publish here is, for example, we were able then to see to locate the mutation, to compare the mutation or variants from different part of the IRES, and then part of our results we wrote in that time in this quite well referenced review. And this is unpublished results. We can also estimate, for example, if you make a mutation or if you find a mutation in some part of the hepatitis C virus, what will be the effect? So, for example, if you make the mutation in 3b subdomain of the IRES, then probably nothing much happened. The decrease will be in general around 80%. If you make the mutation in 3c domain, then most probably you will completely eliminate the function of the IRES. So, and you can, we also were able to estimate what happened if we make multiple mutation if in all these different domains. you know, so you see some decrease, but it keep the how it is. It keep the similar trend as the first one. And then we are thinking of course, if there are so many viral IRESs, are there really, would we will be able to find something like the IRES in the human genome, human transcriptome? And we found that there are no tools to make it in available, really good tools to make it in to make such a big project. So we designed the tool Na2D search. It's unpublished yet, it's still here, but there is it's currently password protected, but we hope we will publish it soon. And we call it nucleic acid secondary structure search suite. It has a nice graphical user interface, and you can either define the descriptor to search the data, the library of structure, the search the structure in a library of structure, the RNA structure or nucleic acid structure. You can define it as a text file, but you can also draw it here. You can put the freedom, some kind of freedom to the loops, to the stems, and so on. You can put the nucleotides there, which you would like to have there, and so on. And this is just in this window example of some results from the model database we search it. This is kind of how we can draw the part of the hepatitis C IRES. This is the full domain 3. So this is the full IRES. This is how it looks like. These arrows show what we would like to draw, and this is the IRES drawn in the Na2D search in the graphical user interface. And then we design a pipeline using this Na2D search tool to search for RNA structures in human transcriptome and human genome. We use different pieces from different IRES structures of viral origin and search for the human genome, human transcriptome for similar structures. Got several candidates because we really put very strict thresholds. So wanted to get only few candidates finally, and were successful in that. And then we made some biological experiments, synthesized the RNA in vitro from these candidates. This is one of them. This is the control. This is sample. And one of the experiments we can do is to do the electrophoretic mobility shift assay, which is here. And we put together the synthesized and folded RNA, this piece of RNA with rabbit reticulocyte translation competent lysate, put it on the gel and native gel, and then we can see some shift when the protein is bound to the band. And when we look to the different concentration of potassium chloride, then we can see in lower concentration, more native concentration, we can see some bands which then disappear in higher concentration. So you can see that the protein is sold out. And currently we cut these bands in some samples, make a mass spec, and trying to identify the proteins and protein complexes there. This is of course not published yet and far to finish. And I would like to thank, because this is, I just summarized a really long part of our studies. So I would like to thank to all current past members of the Laboratory of RNA Biochemistry, collaborators from Institute of Molecular Genetics, University Hospital Ostrava, Institute of Public Health Ostrava, Czech Technical University in Prague, where we are doing the bioinformatics now, and all the funding, including ELIXIR-CZ. So I'm just few seconds before the end. Thank you for your attention, and I'm ready to get your questions.
Martin, thank you very much for a very interesting lecture with a lot of information about the background. We have a few questions. So how intensively are both databases used and who is the target community?
Well, the target community is mostly molecular biologists, virologists who use these databases. Also, there are people who would like to use the IRES or study the IRES in some special model. For example, we are getting questions from the people, so I know this. For example, a few years ago, I got a question about if some IRES which was found in human could work also in some hibernating squirrel, because there is also during hibernation there is not functional normal cap dependent translation. And how it's used, I don't remember. We also reporting in ELIXIR the accesses, so I don't following that. I don't remember that. But for example, the database, the IRESite, these two publications are still well referenced, and we got, I think, a little bit. I look now to Google Scholar. We got more than 200 citations of these publications. So I think, and still is growing, you know. So people are using that, they cite that, and yeah.
So the second question is that two databases are linked internally, or other question is
no, they are separate.
And externally, you're probably using some identifiers from other sources, from other databases. Is that true? So you can actually
yes, we have identifiers from other sources, and we also have in both databases the entries have their own identifier for specific for the database. So you can cite that. And for example, but unfortunately they didn't cite that, but a few years ago when they upgraded RFEM, for example, they wanted to find a lot of proved structures, and there's not that many structures of RNA which are really proved. So we know positively because of the mutation, just batch download all our structures and put them to the RFEM. But we are happy with that. It serves to the community.
Okay, so thank you very much. There is no other question. Martin, thank you again, and look forward to the next Friday where we'll be talking about a CaverDock, which is the tool from Loschmidt Laboratories. Thank you very much and have a good day. Bye.
Thank you. Thank you. Bye.